You know, fungi are literally everywhere, quietly shaping our world. But what if we told you there's this massive hidden kingdom of fungi, and they have this unique jack-in-the-box way of launching spores?
Right, and they're responsible for some major agricultural disasters that
Mm.
actually reshaped farming.
Exactly. And they even give us clues about ancient human history and, modern health issues. Welcome to The Deep Dive, the show where we take complex source material and really pull out the most important nuggets of knowledge and insight for you. I'm your host speaker.
Yeah, I'm your expert speaker. I'm here to help connect the dots, maybe explore some broader implications, and find those aha moments hidden in the details.
Today, we're diving deep into a really fascinating and, frankly, incredibly impactful group of fungi, the Loculoascomycetes.
Hmm.
Our source for this is a detailed chapter from Introduction to Fungi, the third edition, by John Webster and Roland W.S. Weber.
Yeah, a classic text. And our mission today is basically to unpack all that dense science about these fungi.
Mm-hmm.
We want to make it accessible, engaging for you.
So we'll look at what makes them tick, right? Their defining features.
Exactly. Their characteristics, their life cycles, which are pretty diverse, their ecological roles, and their profound real-world significance. We're talking everything from the food on your plate to the air you breathe.
And even archaeology. You mentioned that.
And even archaeology, yeah. It's got a range.
Okay, get ready to understand why these microscopic masters truly matter. Let's unpack this. So let's start right at the core. What's the absolute defining feature? What makes a loculoascomycete, well, a loculoascomycete?
Okay, the thing that really stands out is their unique ascus. That's the sac that holds the spores.
Right.
It's described as bitunicate or fissitunicate.
Right.
Now, picture this. It essentially has two layers, two walls you can separate. There's an outer wall, the ectotunica, and then a thinner inner layer, the endotunica.
Two walls, okay.
Yeah. And here's the cool part. When it's time to release the spores, that outer wall, it doesn't really stretch. It just ruptures, breaks open, maybe at the side or the top, and that lets the inner wall suddenly stretch out, like really rapidly extend, pushing the spores out forcefully, almost like a microscopic water pistol or...
The jack-in-the-box.
Exactly, the jack-in-the-box mechanism. It gives you a really clear visual, doesn't it?
Mm-hmm.
And the key thing here, the insight, is that this forceful discharge lets them shoot spores way further than they otherwise could.
Maximizing dispersal. A real advantage.
A huge evolutionary advantage for colonizing new places efficiently.
That image really sticks. Okay, so these two-walled asci are central. What about the structure they grow inside, their fruit body? Is that special too?
It is. Their fruit body is called an ascostroma. It's basically a mass of fungal tissue.
Okay.
And inside this stroma, you find these cavities called locules. That's where the asci develops. And the development is termed ascolocular.
Ascolocular, meaning...
It means the main structure, the ascoma,
Yeah.
is already formed before the compatible nuclei, the genetic material for sexual reproduction, even come together.
Oh, interesting. So the stage is set before the main event.
Pretty much. Which is different from a lot of other fungi where the fruiting body only starts forming after the nuclei pair up. It suggests a kind of preemptive strategy, maybe more efficient.
That's a really crucial distinction. Are these fruit bodies always the same shape then? Or is there variety?
Oh, lots of variety.
Yeah.
They often form a structure that looks like a flask-shaped perithecia,
Uh-huh.
But technically they're called pseudothecia.
Pseudothecia. Why the difference?
Because the locules, those cavities, are just spaces within the stroma. They aren't surrounded by their own distinct wall like in a true perithecia.
Got it.
But yeah, the diversity is remarkable. They can also be apothecia, sort of cup-shaped, or historothecia, which are elongated with a slit, or even cleistothecia, completely closed spherical things.
Wow. So much variation. It sounds like an incredibly diverse and successful group. Just how big are we talking, numbers-wise?
Oh, it's vast. We're talking about 900 genera, maybe over 7,000 species described so far.
That's huge.
It is, and mostly terrestrial. You find them often as saprotrophs, nature's decomposers, breaking down dead stuff.
Right.
Or as endophytes, living inside plants, often harmlessly, at least initially. But critically, many are also major parasites on plants.
Causing diseases.
Causing diseases, yeah. Often ones with huge economic impacts on agriculture. But you also find them in freshwater, the sea, on dung, in soil.
Yeah.
They're really widespread ecologically.
Okay, this is where it gets really interesting for me. Their evolution. With all that diversity, do they all come from one single ancestor? Are they a neat, tidy group?
Well, that's the thing. Molecular studies, looking at their DNA, suggest that Loculoascomycetes, as we currently classify them, are probably not monophyletic.
Meaning not all from one single common ancestor.
Exactly. Their shared history is more complex. It's likely a collection of groups that evolved these bitunicate asci maybe more than once, or they're related in ways we're still figuring out. They do show close links to other fungi like the perithecia.
Okay. So for our deep dive today, where are we focusing within this complex group?
We'll focus on representatives from two really important orders, the Pleosporales and the Dothideales. They contain a lot of the key players.
Right. Let's start with the Pleosporales then. What makes this order stand out, especially given their impact?
Okay, the Pleosporales. This is a large group, and unlike the broader Loculoascomycetes, molecular evidence suggests this order is mostly monophyletic, so a more closely related bunch.
And they're big trouble for crops.
They include many economically important plant pathogens, yes.
Mhm.
Things like Cochleobolus, Phytosphaeria, Pyrenophora. They often go after crucial crops like grasses and cereals. But it's not all bad news. They also include common endophytes and saprotrophs, too.
And they impact us directly too, right? Beyond just the crops we eat.
Oh, absolutely. Some species in this order, and especially their asexual forms, their anamorphs, can be major human allergens.
Triggering asthma and things like that.
Exactly. Severe asthma sometimes. And some can even be opportunistic human pathogens, causing infections. Plus, crucially, many produce mycotoxins.
Toxic compounds.
Yeah, really toxic compounds that are a serious health risk if they contaminate our food or animal feed.
You mentioned their pseudothecia development earlier, that Pleospora type. Can you unpack that a bit more? What's distinctive there?
Sure. The Pleospora type development is interesting because the fungus sort of builds this internal scaffolding before sex really gets going. Inside the main fungal tissue, the stroma, you get these special vertical threads called pseudoperiphyses growing down into the cavity that's forming.
Like support beams.
Kind of, yeah, creating a network. They asci, the spore sacs, then develop among these pseudoperiphyses at the bottom and grow upwards. And the opening, the ostiole, isn't actively grown. It forms by lisigenous development, basically. Pre-existing tissue just breaks down to make the hole.
Huh. A pre-planned structure. Okay, let's bring this to life. Tell us about Leptosphaeria. You hear that name a lot in plant pathology.
Right, Leptosphaeria. Very widespread. You often find them on decaying leaves and stems doing their saprotroph thing. But yeah, some are serious plant pathogens. The big one is Leptosphaeria maculans.
Causes what disease?
It causes blackleg disease, especially bad in oilseed rape canola and other brassicas like cabbage. It hits the leaves, sure, but the worst damage is usually down at the stem base, and the roots can cause huge losses.
How does it spread?
Well, during the growing season, it's mostly its anamorph, Fomalingum, spreading spores via rain splash. Then it survives the winter on the crop stubble left in the field.
Ah, the stubble is the reservoir.
Exactly. And in spring, the sexual stage, the pseudothecia on that stubble, release ascospores that infect the new crop. It can also be seed-borne, traveling with the seeds.
Nasty. And you mentioned toxins earlier.
Yeah, what's really concerning is that the more destructive strains, the A pathotype, they produce specific toxins. And worse, they can actually detoxify the plant's own chemical defenses, the phytoalexins.
So they disarm the plant before attacking.
Essentially, yes. It's a real biochemical arms race.
Okay, so that's Leptosphaeria. How does something like Alternaria fit in? Because that one seems ubiquitous.
Alternaria is incredibly common, yeah. Many species don't even have a known sexual stage. We only know their asexual conidial form. They cause a huge range of crop diseases, black leaf spot on brassicas, leaf lights on carrots, often seed-borne as well. And they have those distinctive spores, right? They do. Many Alternaria, like A. brassicae, produce these beaked conidia. They have these long, thin extensions.
Not just for looks, presumably.
Definitely not.
Right.
That beak significantly increases their chances of getting picked up by the wind, and it reduces how quickly they fall. So they travel further. Great for dispersal.
Clever adaptation. But they're also a problem beyond plants.
Big time. Alternaria species produce a whole suite of mycotoxins. If these get into human food or animal feed, they can be severely toxic. And then there's the direct health impact.
Allergies again.
Major cause of inhalant allergies. Alternaria spores are abundant in the air, especially late summer or early autumn. They're a huge trigger for allergies and, yes, severe asthma. Plus, some can even be opportunistic human pathogens causing infections. They're just everywhere.
Okay, speaking of really serious pathogens, let's talk about Cochleobolus. This group has a truly dramatic history, especially with staple crops like corn and oats. What's their deal?
Yeah, Cochleobolus. This genus contains some of the most damaging plant pathogens ever studied. And a lot of it comes down to toxins. They produce incredibly potent often highly specific toxins that really dictate which plants they can attack and how badly.
Specificity is key then.
Absolutely.
Yeah.
Take Cochleobolus sativus. It causes root rots and leaf spots in cereals. It uses toxins like prehelminthosporol to weaken the host, and it can even evade the plant's natural defense mechanisms, like the oxidative burst.
But the most dramatic stories, the real turning points, involved specific outbreaks, didn't they? Like what happened with Victoria oats.
Exactly. This really hammers home the specificity point. There was an oat cultivar called Victoria, bred specifically because it was resistant to rust disease.
Okay, sounds good so far.
It was, until Cochleobolus victoriae showed up. This fungus produced a toxin called Victorin C, and this toxin was so incredibly potent and specific to Victoria oats that just applying purified Victorin to the plants could reproduce all the symptoms of the disease, caused massive cell death.
Wow, a single toxin doing all that damage.
Yeah, and interestingly, it seems to act on the plant's mitochondria, kind of like inducing apoptosis or programmed cell death in mammals.
And then there's the infamous 1970 U.S. corn epidemic. That was Cochleobolus too, right? A watershed
A
moment.
huge one. That was Cochleobolus heterostrophus and its T toxin. Here's the background. Maize breeders, to make hybrid seed production easier, had become heavily reliant on corn varieties with something called Texas male sterile cytoplasm. These plants had a specific mutation in their mitochondria,
Okay, convenient for breeding.
but a massive vulnerability waiting to happen. A new race of C. heterostrophus, race T, emerged, and it produced this T toxin,
Which
specifically targeted those mitochondria with the Texas male sterile mutation. It made the mitochondria leaky, disrupting energy production, killing the cells. The result? Widespread southern cornleaf blight. Over $1 billion in crop losses in 1970 alone. It was catastrophic.
$1 billion just from one fungal race targeting one genetic trait.
Exactly. It was a brutal lesson.
So this wasn't just an agricultural disaster, it was a huge wake-up call, wasn't it? What was the single biggest lesson the scientific community learned from that about, crop resilience and breeding?
Absolutely. The biggest takeaway. The sheer danger of genetic uniformity, of monoculture, relying so heavily on that one mitochondrial trait for male sterility, created this enormous Achilles heel across millions of acres.
So diversity became the watchword.
Precisely. It drove home that genetic diversity in crops isn't just nice to have, it's absolutely essential as a buffer against rapidly evolving pathogens like these fungi. It fundamentally changed modern plant breeding strategies. We learned the hard way.
And it gets even wilder, evolutionarily speaking.
It does. Studies of the genes involved, like mating-type genes, suggest that C. victoria, the oat pathogen, might have actually arisen from a different Cochleobolus species, C. carbonum, which infects corn,
Oh.
through horizontal gene transfer. Essentially, it looks like it might have acquired the genes needed to attack oats from a completely different organism, maybe another fungus or bacterium, instantly changing its host range.
Wow, like swapping genetic toolkits.
Yeah.
A rapid evolutionary jump.
Exactly. It highlights how quickly new aggressive pathogen strains can potentially emerge. These fungi are constantly evolving.
Okay, let's shift gears slightly, but stay within the Pleosporales. What about Pyrenophora and Venturia? They also cause major crop diseases.
Right. Pyrenophora triticea repentis, for example, causes tan spot or yellow leaf spot on wheat. Like many others, it overwinters on stubble, which is the main source of infection in the spring. Its weapons are also toxins, but interestingly, they're unusual extracellular proteins that seem key for a host specificity.
Proteins, not small molecules.
Yeah. And then there's Venturia inequalis. Everyone who grows apples knows this one.
Apple scab.
The notorious apple scab, a serious disease worldwide. And it has a really unusual life cycle for this group. It infects living apple leaves, okay, but the fungus mostly just grows in the tiny space between the leaf's outer layer, the cuticle, and the epidermis below.
So it doesn't immediately invade deep?
Not really. It only goes deeper and becomes more invasive after the leaf dies and falls in autumn. Then it switches gears, becomes saprotrophic, lives on the dead leaf tissue, and produces its pseudothecia there to release spores the next spring.
That's a very specific cycle. Makes it a target for management, I suppose.
Definitely. A lot of apple scab management focuses on stopping those spring spores from the overwintered leaves. This involves disease forecasting, knowing exactly how long leaves need to stay wet at certain temperatures for infection to happen.
So growers can time their sprays.
Precisely. Using protective and curative fungicides. Plus, strategies to reduce the amount of fungus surviving the winter, like treating fallen leaves with urea to speed up decomposition, or shredding them. And of course, breeding resistant apple varieties is a long-term goal.
Okay, one of the most surprising things I read was how a fungus could act like a microbial historian, telling us about ancient history.
Ah, yes. That brings us to Sporamiella, a fascinating group. They have these very distinctive, dark, multi-celled ascospores. Often they have a characteristic germ slit where the germination tube emerges. And the spores can even break apart into individual cells that can still germinate.
Okay, distinctive spores. But the history part.
The key is where you find them. Sporamiella species are almost exclusively found growing on the dung of herbivores, plant eaters.
Dung fungi. Got it.
Exactly. And because their spores are so unique and easily recognizable, and so strongly linked to dung, they've become this amazing tool in archaeology and paleoecology. They're like a fungal fossil record of large animal populations.
How does that work? Give us an example.
Okay, the classic example is from Madagascar. Scientists looked at sediment cores, like layers of mud built up over centuries in lakes or bogs. They counted the Sporamiella spores in each layer, and they found the abundance of spores directly tracked the presence of large herbivores. There was a dramatic decline in Sporamiella spore density starting around 200 AD.
Which coincides with?
Exactly when humans first settled Madagascar, and tragically, when most of the island's unique large animals, the megafauna like giant lemurs and elephant birds, went extinct. Fewer large herbivores meant less dung, meant fewer Sporamiella spores.
Wow. Direct evidence in the mud.
But then, after about 1100 AD, the spore density started to increase again.
Wow.
That lines up perfectly with the time humans introduced domesticated livestock, cattle, sheep, goats, to the island. New herbivores, new dung, Sporamiella bounces back. It's an incredible fungal record keeper of ecosystem change and human impact.
That is genuinely amazing. Okay, let's shift focus now to the other major order you mentioned, the Dothideales. How do they compare with the Pleosporales? What sets them apart?
Right, the Dothideales, another huge group, also with those bitunicate asci. But a key difference, generally speaking, is that they lack that internal scaffolding, the interascal tissue or pseudoperiphyses that we talked about in the Pleosporales.
So the asci develop more freely in the locule.
Often, yes. They showed just enormous variety in their asexual forms, their conidia, and in their ecological roles, too. Lots of saprotrophs, lots of plant pathogens again. Just incredibly diverse lifestyles.
And Mycosphorella is a really big player here, isn't it?
Oh, absolutely huge.
Yeah.
Mycosphorella is one of the largest genera of all ascomycetes, maybe over 2,000 species described. Many cause really significant plant diseases, and tissue death, necrosis, is a common symptom. And often the toxins they produce are directly linked to that damage you see on the plant.
Give us an example.
Well, Mycosphorella graminicola causes septoria leaf blotch of wheat, very damaging disease. Its ascospores, released from pseudothecia on that overwintering stubble, sound familiar, are the main source of infection.
Same strat.
Same basic strategy. And it leads to incredible genetic diversity in the field. You might find like 70 different genetic strains in just 1 square meter of a wheat field.
Wow. Constant evolution.
Constant pressure. Interestingly, M. graminicola almost always infects the leaf by growing in through the stomata, the leaf pores, unlike some others that can push directly through the cuticle.
Okay. Tell us about Cercospora. You mentioned this one involves a particularly interesting toxin, something about light.
Yes, Cercospora, another massive genus of plant pathogens, leaf spot of sugar beet, gray leaf spot of corn, lots of others. What's really unique is their toxin, cercosporin. It's a photosensitizing toxin.
Photosensitizing, meaning light activates it.
Exactly. When cercosporin absorbs light energy, it gets activated, and it reacts with oxygen molecules in the cell, transforming them into highly reactive forms, particularly singlet oxygen.
Which is bad news for the cell.
Very bad news. Singlet oxygen is incredibly destructive. It just rips apart organic molecules, especially the lipids that make up cell membranes. They just fall apart.
So that's why the damage happens.
And that's why Cercospora infections are way less severe on plants grown in the shade compared to plants out in full sun. More light means more activated toxin, means more damage.
Fascinating. How does the fungus not poison itself?
Good question. It seems to have ways to protect itself. Maybe by keeping the toxin in a non-reactive state until it's outside its own cells, or by producing antioxidants like vitamin B6 to neutralize the reactive oxygen.
Clever biochemistry. Now, what I found really striking was how that common fuzzy mold, the one you see everywhere, is actually part of this Dothideales group.
Ah, you must be talking about Cladosporium. Yes, species like Cladosporium herbarum are truly ubiquitous. They are one of the most abundant fungi you find in the air spore, the spores floating in the air.
So that dusty, greenish black stuff.
That's often it. It's a frequent contaminant on food, damp surfaces, even in labs. It's practically everywhere.
And it's not just a nuisance, right? It's directly linked to our health.
Very directly. Cladosporium herbarum, along with alternaria, which we mentioned earlier, is strongly associated with severe asthma and mold allergies. There are over 30 different components, antigens, from Cladosporium that are known to trigger allergic reactions in people.
30? Wow.
Yeah. You recognize their colonies, dull, olive green to black, and microscopically, they produce their conidia, their asexual spores, in these distinctive branching chains.
There's another cladosporium that's interesting too, C. fulvum. It's different, right?
Yes, Cladosporium fulvum is unusual for this group because it's a biotrophic pathogen, specifically of tomato plants.
Biotrophic meaning it lives with the host without killing it immediately.
Exactly. It grows its hyphae, its fungal threads, in the spaces between the tomato leaf cells, interacting closely but usually without forming specialized feeding structures called haustoria. It even messes with the plant's sugar transport, converting sucrose into mannitol for its own use, which causes those characteristic yellow chlorotic spots on the leaves.
And this one has been important for understanding plant defenses.
Hugely important. The interaction between C. fulvum and tomato is a classic textbook example of a gene-for-gene relationship.
Explain them.
Basically, the tomato plant has specific resistance genes, R genes. The fungus has corresponding avirulence genes, AVAR genes. If the plant has the R gene that recognizes the protein produced by the fungus's AVAR gene, it triggers a strong defense reaction, often a hypersensitive response, where the plant cells around the infection site die quickly, stopping the fungus in its tracks.
So a molecular lock and key.
A perfect analogy.
Yeah.
And studying these specific gene interactions has given us critical insights into how plants recognize pathogens and activate their immune systems. Research is still ongoing.
Okay, finally, let's touch on Oreobasidium and this intriguing group called black yeasts.
Right. Oreobasidium pullulans, another one that's just everywhere. Ubiquitous saprotroph, plant surfaces, soil, water, air, even sewage treatment plants.
Then it's a shapeshifter.
It is. It's pleomorphic, meaning it can switch between different growth forms. It can grow as typical fungal mycelium, those long threads. Or it can produce these slimy masses of yeast-like cells called blastoconidia that reproduce by budding. Or it can form thick-walled resting spores called chlamydospores. It adapts its form to its conditions.
With all that adaptability, does it have any practical uses, or is it just there?
Oh, definitely potential uses. It's being seriously investigated as a biocontrol agent, meaning using it to fight other fungi, specifically fungi that cause fruits to rot after harvest, like on grapes and strawberries. Oreobasidium can outcompete them or inhibit their growth.
A natural preservative, almost.
Kind of. It's also a source of industrial products like gluconic acid and something called pullulan.
Pullulan.
It's an extracellular polysaccharide, a complex sugar it secretes. It's used as an adhesive in special films and fabrics, and even as a low-calorie ingredient in foods and pharmaceuticals. So potentially quite useful.
But you mentioned black yeasts. Not all of them are so beneficial, are they? Some sound quite dangerous.
That's true. Black yeast isn't a strict taxonomic group. It's more a descriptive term for fungi that produce dark, melanized yeast-like cells. And yes, some of these can be nasty, opportunistic human pathogens.
Like what?
Genera like Ixophiala and Cladophyalophora contain species known to cause really severe, sometimes fatal, infections, particularly in people with weakened immune systems, brain infections, skin infections, systemic infections.
And are these related to the Dothideales we've been discussing?
Historically, some were thought to be, with new molecular data. Many of these pathogenic black yeasts are now classified in completely different fungal orders, often in the Eurocheomycetes.
So not strictly Loculoascomycetes anymore.
Not strictly, no. But ecologically, they often share similarities, frequently found on decaying plants and in soil, like many Dothideales. It highlights how complex fungal relationships are and how our understanding keeps evolving.
Okay, wow. So what does this all mean? We've taken a really deep dive here into this fascinating, complex, sometimes scary world of Loculoascomycetes. If you had to boil it down, what are the absolute essential takeaways for our listener?
Right, connecting it all back, the big picture is Loculoascomycetes, defined by that unique bitunicate ascus that Jack in the Box, are an incredibly diverse and really impactful group of fungi. We've seen just a huge array of forms, both sexual and asexual.
And their impact is just enormous, isn't it? From, entire agricultural systems down to our individual health.
Absolutely. Ecologically, they're everywhere. Critical decomposers, sure, but also hugely significant plant pathogens causing immense economic losses, forcing us constantly to develop new management strategies. That whole biochemical warfare aspect is key.
And the direct effects on allergies, toxins in food.
Exactly. Major allergens floating in the air we breathe, potent mycotoxins contaminating food and feed, and even acting as opportunistic human pathogens causing serious infections. They hit us from multiple angles.
And scientifically, they've taught us so much.
Definitely. The insights they've provided into basic biology, host-pathogen interactions, the gene-for-gene concept, even things like horizontal gene transfer. These are fundamental principles. Studying these fungi has revealed deep insights into the evolutionary arms race between organisms. These tiny things teach big lessons.
So what does this all mean for you, our listener, as you go about your day, maybe see a patch of mold, eat an apple, breathe the air? Well, given their incredible adaptability, their mastery of chemical warfare with these potent toxins, and their constant evolutionary shifting, it really raises a provocative question, doesn't it? What other undiscovered roles might these Loculoascomycetes be playing right now? In ecosystems, maybe in medicine perhaps, undiscovered antibiotics or enzymes, or even presenting completely new challenges in agriculture or health that we haven't even imagined yet. Keep an eye on these tiny, mighty organisms. They are shaping our world in ways we often don't see, and you can be sure their story is far from over. And that brings us to the end of another deep dive. Thank you so much for joining us on this fascinating exploration of the Loculoascomycetes.
Yes, thank you. We really hope this diet has given you some valuable insights, maybe sparked your own curiosity to learn more about this incredible fungal kingdom. There's always more to discover.
Until next time, stay curious and keep diving deep.